Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Jordan Stillman, Project Coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. Please enter your questions at any time. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you again for joining us today. It is now my pleasure to introduce our speaker, Ben Josey from Corning Life Sciences.
Thank you, Nicole. Hi, everybody, and thank you for inviting me to speak here today. The focus of this brief talk will be to introduce an interesting new technology from Corning for separating and processing cells in an efficient reagent-free closed system. Next slide. The system that I'm referring to is Corning's new X-Series cell processing platform. Today, I will be discussing two components of this system, the X-Lab and the X-Wash. I will briefly go over some of the background and history about the different types of cell separation technologies in use, and then discuss how Corning's new technology brings advancements to this field. I will then do a bit of a deep dive into how this technology works, followed by some presentation of some data from a variety of applications that these devices can be useful for. Now, I'm sure that the majority of this audience has experience with cell separation in some format, whether it be simple separations of cells from media as a part of basic cell culture practices, buffer and media exchanges, or precise selections of specific cell populations. All of these methods can be broadly divided into four categories: filtration, centrifugation, affinity purification, which is typically an antibody-based capture, and other, which includes unique technologies that are still mostly in the development stages. If you focus on the first three columns, generally speaking, as you move from left to right, from filtration to affinity purification, and then again looking at the subcategories, what you deal with is trying to strike a balance between the precision with which you can isolate specific cell populations and the batch size that you can process efficiently in terms of things like time, labor, and cost. Another major consideration, especially in the therapeutic space, is the addition of extraneous reagents into this milieu. Corning's X-Series systems fall into the centrifugation category. But add to this an intelligent sensor control system that provides a high level of flexibility and automation over how the cells move through the single-use cartridge during processing. I will get more into the details of how this works in the upcoming slides. In the cell therapy space, blood and other blood products like aphereses, cord blood, bone marrow, etc., are common starting materials. Oftentimes, only a specific component of the blood is desired for downstream processing, and thus there is a need to separate the blood into its various components, often going so far as to progressively select out very specific cell types. The traditional method of processing blood is based on centrifugation, which takes advantage of the different densities of each of the blood cell types and then applies G-force to them to accelerate their stratification into different density layers. Similar to what you might have observed in those childhood experiments where the teacher would mix differently colored oil and water together and then they would separate out into layers that you could see. A traditional method of increasing the precision of this separation, for example, in applications that need to specifically isolate mononuclear cells, a density gradient media, such as Corning's lymphocyte separation media, or the more common household brand name, the Ficol reagent, is added to a tube, and then the blood is carefully layered on top. The density of this media is chosen to sit between the densities of the cells of interest, so that after centrifugation, some cells will move below the media and others will be trapped above it. Now, this Ficol-based method has served its purpose for many years, but it does have several drawbacks. Aside from the need to add that additional reagent, this is a tedious, labor-intensive manual process with a high degree of sample and operator technique-based variability that can result in reduced yields and viability. Corning's XLab system seeks to provide a solution to this issue in a convenient format compatible with a swing bucket centrifuge rotor. The process can range from automated to semi-automated depending on the user's desires and can handle up to 240 milliliters per single-use cartridge. The device is registered as a Class 1 medical device, and it can enable full 21 CFR compliance. And you've heard me toss around the terms XLab and XWash. To alleviate some confusion there, the XLab and the XWash systems are really very similar, with the primary differences being in the design of the disposable cartridge to better suit them for different applications. These cartridges consist of multiple valve-gated chambers into which different cell populations are routed. In addition to the cartridge, both systems also include a reusable control module that opens and closes the valves in the cartridge, and a docking station that charges and processes data from the control module. Now, this control module is really where the core of the system technology lies. This unit connects to the base of each disposable cartridge and contains four main components. A tower of optical sensors that you can see here in the upper right-hand image, indicated by the yellow dots. These sensors monitor and detect the cell layers during centrifugation. It also contains an accelerometer that monitors the G-force and cams that open and close chamber valves in the disposable cartridge. Onboard memory also continuously records process data such as time and the inputs from the other components. Now, all of that works together to allow you to precisely move fractions into discrete chambers within the disposable cartridge. The cartridge design is the primary differentiating factor between the XLab and XWash systems, with the main differences being application-specific designs in the tubing accessory for loading and sampling, and the number of chambers, with the XLab having 3 chambers and the XWash having 2. Whereas the XLab system is designed primarily for separating out individual cell fractions, the XWash system is tailored more for applications such as cryoprotectant removal, cell washing, volume reduction, and such. The tubing configuration allows for multiple media changes to be performed, and consistently high recoveries and viabilities for a variety of cell types have been observed. So what does a typical workflow using one of these devices look like? Well, you start by loading your sample into the cartridge. This can be done using either spike ports or sterile tube welding. The cartridge is then attached to the control module base and loaded into the centrifuge, where the control module will execute its preloaded protocol. This process can be fully automated, or it's possible to interrupt it to perform additional steps such as media exchanges or resuspensions. After the process is complete, you just withdraw the harvested cells using either the provided Luer lock connection or, again, by sterile tube welding, and then proceed to download the process report. A typical PBMC isolation from whole bud will take about 35 minutes, so this is not a fixed time. You can switch some things up to make it run a little bit shorter. And what will happen during this process is that sequential centrifugation steps at various G settings will stratify the cells. As these cell layers form with their different densities, sizes, and optical properties, the sensor tower will detect these layers and communicate with the cams to open and close valves in the cartridge to route different cell populations into different chambers within the cartridge. So now I'm just going to briefly run through some data for a few of the most common applications that the X-Series systems can be used for. A super common process is the isolation of mononuclear cells from peripheral blood or bone marrow. As you can see in this data here, This process consistently provides greater than 90% recoveries of target cell populations with significant depletions of unwanted cell types such as the platelets and granulocytes. An important thing to highlight here, and a particular strength of this system, is its ability to consistently provide greater than 99% red blood cell depletion. And it does this without the need for an RBC lysis step. This is truly impressive compared to any other method that I've seen. Now, if you need to concentrate a sample or otherwise perform some kind of volume reduction, the system is also very capable of doing that, from volumes up to 240 mils, concentrating all the way down to 3 mils. Washing and buffer exchanges are also very effective, with a high percentage of media exchange occurring while retaining greater than 90% recovery and viability in the cells. You can see here this data is performed with just some Jurkat cells. However, we've seen it repeated multiple times with different cell types, and you can obtain similar results. Another common application of interest is the removal of DMSO or other cryoprotectants from thawed-celled products. Here, you can see some data from processing frozen leukopaks obtained from 3 separate donors, in which greater than 99% of the DMSO was able to be removed from 2 wash steps, with negligible losses in the recovered cells occurring. Even with only 1 wash step, I'm not sure if you can see the numbers, the DMSO concentration was reduced by greater than 95%, which for most applications is likely to be more than sufficient when you take the subsequent dilution and formulation steps into consideration. Now, one more important thing to highlight here is the traceability that this device provides in terms of 21 CFR compliance and any GMP reporting that you might want to do. As I previously stated, this is registered as a US Class 1 medical device, and that control module is continuously monitoring and recording during the entire process. At the end of the run, you're able to download that data, and the software will generate a report that looks something like what you see here. That software, it allows for tiered access with administrator level, supervisor level, and technician level access, as well as a tiered review process for these reports. Each of the disposable cartridges comes with a barcode tracking system. And you can see several of the process parameters kind of here in this graph that lets you know at what G-force, the amount of time spent at each step, and indicators of whether or not each particular step of your pre-programmed process was performed as indicated. Now, just to summarize a bit of what we just talked about, I introduced the new Corning X-Series cell separation platform. With this platform, we are able to consistently achieve high-quality results with MNC recoveries typically greater than 90%, and this MNC enrichment process occurring in approximately 35-minute times, sometimes less. A really important thing to highlight power of this system is its ability to deplete red blood cells. We consistently see red blood cell depletion in greater than 99% without the need for an additional lysis step of any kind. It's an automated, sterile, closed system, which provides reliable collection of all of the fractions which are routed into the separate chambers. Things like mononucleated cells, red blood cells, and plasma can all be separated reliably in a reagent-free closed system. We see high viability, recovery in cell washing and buffer exchange processes, and it has the ability to concentrate down a sample all the way down to 3 mil volume, which I know for some people, getting that harvest volume down as low as possible is really important. And importantly, for GMP compliance, everything that happens across the process is tracked in real time by that control module. And this allows for the generation of tiered report reviews and batch reporting. And with that, I'd like you to please see the URL presented here next to this nice graphic. And this can take you to a web page where you can learn more or request a virtual demonstration. And with that, I'd like to open the floor for some questions.
Fantastic. Thank you so much, Ben. All right, so we have a couple questions. The first one here is, what is the smallest sample size that can be processed?
Oh, that is a good question. So we state that we can process sample sizes in the range of 40 milliliters all the way up to 240 milliliters, and that is very possible. But we find that you get better results if you increase that, just dilute that small sample up to about 60 mils. So if you're dealing with a small sample size, you can add a little bit of buffer media or even just PBS in there and bring that sample volume up to 60. But you do have the capability of running down as low as 40.
Fantastic. And here's another question. What if I am not interested in the MNCs, but instead would like to isolate a different cell population? Is that possible?
Ooh, that is another good question. Yes, absolutely. This system is incredibly flexible. Now, for a lot of the GMP compliance parameters, it's important to note that once that control module is programmed, it's not a simple thing to just go in there and have your technicians modify it. So it's really locked in and kind of an almost plug and play from the user perspective there. However, as I showed you the parameters in that control module with the tower of optical sensors and the accelerometer and the cams, that really gives a high degree of flexibility in how you want to control the movement of those cells through the cartridge. And by playing around with things like differential centrifugation, doing different G-speed spins, you can ramp up the speed, ramp down the speed, change the gain settings in the sensor detectors. You can really tailor this system to isolate, to stratify and move cell populations pretty nicely. And the way you access these is by those multiple chambers in the cartridge. So the XLab system, for example, it has 3 chambers in there. So you can really get 3 fractions out of it. You've got the main chamber where you initially load your sample. In a typical PBNC isolation process, at the end of that process, you're going to have either platelet-rich or platelet-poor plasma contained in the main chamber. Then you also have a depletion chamber, which will contain primarily your red blood cells and your granulocytes. And then you will also have your harvest chamber, which will contain your mononuclear cells. So yes, it's definitely flexible.
Fantastic. Well, thank you so much, Ben. I think that's all the time we have for questions today. Thank you so much for joining us. And I would also like to say, fantastic. And I'd also like to say thanks to our audience for joining us today as well. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.